So next very important topic is enzymes. Okay, so before going to enzymes proper, you have to know the introduction about enzymes I'll tell. Here in enzymology, important thing that you have to study is about competitive and non-competitive inhibition, also about the structure of enzyme and also the classification of enzyme, then about the various mechanism of enzyme action. So first of all, what are enzymes? So enzymes are nothing but they are group of biomolecules synthesized by living cells. So group of biomolecules synthesized by living cells are known as enzymes. They are catalysts or biological system. I mean they are biocatalysts. So for understanding enzymes, remember, suppose you are traveling to a certain place. So you can go by walking, you can go by taking a bike, you can go by taking an auto, bus, etc. But suppose you are going to a far away place, going by aeroplane is easier. So that is an enzyme for that mechanism. So enzymes makes mechanism easier. Okay, so they are catalyst of biological system. That's why they are known as catalysts for making an mechanism easier. Okay, now they speed up. Main function is they speed up chemical reaction. So they are colloidal, tiny particles. They are thermolabile because they are heat sensitive. They help in converting one form of energy into another. So decide how, when chemical changes happen in the body. Now key features of enzymes are mainly, there are basically three key features are there. First one is catalytic power. Enzyme has a catalytic power. They have a specificity. Enzyme has a specificity and regulation. See, enzymes make chemical reaction happen faster, even 1,000,000 times faster. That's why it has a catalytic power. They do this by lowering the activation energy. So activation energy they will lower and they will speed up the that activation energy means minimum energy needed to start a reaction. So in our body, temperature will be always stable. So enzymes helps reaction happen without needing to raise the temperature. Okay, that is known as catalytic power. Each enzyme works on a specific substance called substrate, right? So you know about the enzyme substrate complex. See, this is an enzyme. So enzyme will have an active site. This is the particular thing, like works on a specific substance, this substance. So that's how they act. And end product, enzyme substrate complex will be formed. So like a key fills on a lock. That's how the mechanism happens. Now third one is regulation. The activation enzyme of an enzyme is carefully regulated by body to keep everything in a balance. Now most of the enzymes are proteins. Okay, a few special enzymes called riboenzymes are made up of RNA, not protein. Remember, so riboenzymes are proteins. They will give some questions like this, true or false. So riboenzymes are not protein. They are RNA, made up of RNA. Ribonucleic acid. Some enzymes are just proteins while others have a protein part plus a non-protein part. They are known as conjugated protein part. In such enzyme, certain enzyme will have a protein part and a non-protein part. Okay, so this non-protein part is known as prosthetic group or coenzyme. The non-protein part is called apoenzyme. So together, what you call it is known as holoenzyme. Holoenzyme is nothing but the apoenzyme plus and coenzyme is known as holoenzyme. Once more I'll tell you, some enzyme will have a protein part and a non-protein part. So this protein part is known as prosthetic part, that is coenzyme. I mean non-protein part is of course prosthetic part, that is coenzyme part. Protein part is apoenzyme. Remember, apoenzyme plus coenzyme is holoenzyme. Okay. Now there are, in general, there is a categorization of enzyme, that is this one, 6 classes of enzyme. Before going to that, there is another classification that you should know, that is based on the peptide chain and all. So there is one classification, that is monomeric enzymes. Only one polypeptide chain will be there, that is known as ribonuclease. Then oligomeric enzymes are there. Oligomeric enzyme. Oligomeric enzymes contains more than one polypeptide chain. Example, lactate dehydrogenase and hexokinase. So each single unit of this polypeptide chain of oligomeric enzymes are known as subunit. Now you have something called as coenzyme. I told you coenzyme is the non-protein part of a certain enzyme, require a specific, so some enzyme require a specific thermostable low molecular weight non-protein organic substance. So it is a thermostable low molecular weight organic substance. So a coenzyme binds covalently or non-covalently to an apoenzyme. So this include oxidoreductase, then isomerization and covalent bond form require coenzyme. Just remember what is a coenzyme and examples, that's it. So coenzyme, co-substrate or a second substrate. It is also known as a co-substrate or second substrate. So I tell you what are the coenzymes of certain enzymes. First one is vitamin B complex pantothenic acid has a coenzyme called CoASH. Okay, then vitamin B12 has a coenzyme called cobamide. Folic acid has a coenzyme called as tetrahydrofolate. Niacin has a coenzyme called as NAD plus NADP. Riboflavin has a coenzyme called FMN, FAD, like that, etc. Now 6 classes of coenzymes you have, that is of OT-Hill. Remember like this, nomenclature or classification of enzyme, OT-Hill. Under that you have oxidoreductase, transferase, hydrolases, liases, then isomerases. Oxidoreductase means that include alcohol dehydrogenase, LDH, xanthine oxidase, glutathione reductase, etc. So oxidoreductase include LDH, alcohol dehydrogenase, xanthine oxidase, glutathione reductase, glucose 6-phosphate dehydrogenase. This is involved in oxidation and reduction reaction. Transferase include aspartate, alanine transferase, hexokinase, phosphoglutamate, etc. Hydrolase means that bring about hydrolysis, glucose 6-phosphate, pepsin, trypsin, etc. Then liases include fumarate, and then that lysis means removal of small molecule from a larger substrate. Okay, fumarate is histidine decarboxylase. Isomerases include UDP glucose, then epimerase, racinases, triose, phosphoisomerase. Okay. Then ligases include enzymes involved in joining a substrate, that is RNA synthase, glutamine and DNA ligase. Next one is